Hello and welcome. Today we will be discussing recent understandings in relation to iron metabolism in dementia. Let's first discuss what is dementia. Dementia has symptoms that vary according to the progression of the disease. In the early stages, symptoms may manifest as faint memory loss. As the disease progresses, the level of memory loss experienced by an affected individual continues to increase. Examples may include forgetting names and recent events, to forgetting the names of loved ones at its later stages. The level of dependency on others at the later stages is quite high. Dementia affects over 50 million people worldwide, accounting for the real per capita health and social expenditure increase that occurs at 1% annually. The real cost of dementia is predicted to increase from 14.2 billion to a total of $47.8 billion by 2050. The exact cause of the syndrome appears to be unknown. However, early dementia diagnosis is said to enable the detection of reversible causes of cognitive impairment, maximize patient autonomy, promote patient safety, and allow for the possibility of pharmacological management and engagement in clinical trials. Considering this alongside the financial burden it has on society, finding a solution to alleviate its cause is imperative. Recent studies have suggested that the dysregulation of iron metabolism may impact the development of conditions that cause dementia. Iron plays a fundamental role in mitochondrial function and the transport of oxygen. Whilst iron is essential, the lack of a proper excretion system results in iron being recycled by aging red blood cells, subsequently being converted into compounds such as ferritin and transferrin. Recent case studies have noted that patients with susceptibility to excess iron presence, particularly those afflicted with conditions such as hemochromatosis, are likely to experience significant cognitive decline and rapid clinical deterioration that is not usually observed in dementia-causing diseases such as Alzheimer's disease. Such studies have also suggested that iron content is noted to increase with age, generating the hypothesis that changes in genes regulating iron metabolism acts together with acquired endogenous and environmental factors in the pathogenesis of Alzheimer's. An example of such endogenous factors may be in relation to hepcidin, a regulatory hormone which serves to reduce cellular iron transport by binding to ferroportin, a transporter protein that takes iron from within a cell to systemic circulation. Disturbances to hepcidin function, which may take place genetically, such as through hereditary hemochromatosis, or by disrupting cellular signaling between iron and hepcidin, a mechanism that occurs as a result of mutations in upstream regulators of iron signaling to hepcidin. It may also occur by way of disruption of the hepcidin gene, HAMP, and disruption to the hepcidin ferroportin axis mechanism may also occur by way of pharmacological intervention. A very recent concept uncovered with respect to iron metabolism is ferroapoptosis, essentially iron-dependent cell death that is brought about by glutathione depletion and reduced glutathione peroxidase 4 activity. Glutathione peroxidase 4 metabolizes lipid peroxides, and with reduced activity in this regard, iron oxidizes lipids to create more free radicals that is known to cause further damage to tissue, especially neuronal tissue. The specific regulatory mechanism of ferroapoptosis is still unknown. However, the scope of its operation is largely genetic, with specific molecules and genes such as erastin, RSL3, and DPI7 being known to deplete or degrade glutathione peroxidase 4 activity. Neurological diseases such as Alzheimer's disease are frequently associated with high iron levels and low glutathione 4 and glutathione activity. Accordingly, inhibiting ferroapoptosis in neurons has been suggested to effectively improve the prognosis in dementia-inducing diseases such as Alzheimer's. Further studies do need to occur, however, to definitively monitor the specific mechanisms involved. In light of the aforementioned principles and studies examined, the only conclusions we are able to draw at this present time are that dementia may originate from a range of diseases. Excess iron has been associated with neurological dementia-inducing diseases such as Alzheimer's, and much more investigation is to come with regard to isolating the question of whether iron metabolism is a definite cause of dementia. Thank you very much for watching.